in the RCTs, and they found that it held in adults. They found it held in children, although it's based on a single study, and there's been some controversy about that study. But interestingly enough, they also looked at, these are trials where the low-calorie sweetener was substituted for a nutritive sweetener. Down here are trials where the low-calorie sweetener is compared to water. And they show a beneficial effect of low-calorie sweeteners relative to water, driven largely by the John Peters study out of Colorado, the most recent trial reported in the literature. Nevertheless, so among these three studies, a beneficial effect. And then we get this trial by Azad and group published in the Canadian Medical Association Journal. I just don't know what to make of this trial. They claim to have searched the same databases. They claim to have used common search terms like low-calorie sweetener and obesity, and yet published 2 or 3 years after the other two trials, which each found 13, 15 studies or so, not a single overlapping paper in this meta-analysis with the other two. You just have to wonder how that is at all possible. Additionally, you really have to wonder where some of these studies came from. So, for example, this Gagnon trial, which showed a positive association with BMI, well, if you go back and read the paper, they only looked at individuals who maintained or gained body weight during the observation period. They excluded anybody that may have lost weight. So it was a biased sample to even begin with. It's no surprise at all that it showed a positive effect. It was designed to show a positive effect among these trials. So all three of these data points came from a single study. I have no quibbles with the study itself. It was done here by Harvard, Frank and Walt and Eric Rim and so on. But it was a paper designed to explore different models for predicting effects of lifestyle on weight change over long periods of time, and it looked at things like exercise and other factors. I really don't think it was designed at all to isolate the effects of a low-calorie sweetener on body weight. Nevertheless, they reported a positive association between use of low-calorie sweeteners and BMI, and this captured a lot of media attention once again. And when they looked at the randomized control trials, in contrast to the other two meta-analyses, they found no effect. This is a particularly interesting analysis. 2 out of these 3 trials gave stevia in capsules with the primary outcome variable being blood pressure. So not designed at all to be measuring effects on body weight. It strikes me, and I'm not going to go on bashing meta-analyses, but it strikes me in an era where we are so concerned about registering trials to make sure that the primary endpoints reported in the paper were the a priori identified outcomes of the paper, that we don't use that same criteria for allowing papers to be entered into a meta-analysis. If you're looking at the effects on body weight, it seems the paper in that analysis ought to have been designed to measure body weight. But that's not the case up here. And in this analysis, looking at non-nutritive sweeteners and body weight, this one trial by Tate, let's see, I'm drawing a blank on, at any rate, suffice it to say, I have my issues with this analysis. I really don't know how to put it in the context of the greater literature. Okay, well, so that's kind of where we stand, and what have the professional bodies done with this evidence base? So those that would have a particular interest in the use of patients using these products, so American and Canadian Diabetes Associations, Heart Association, the Academy of Nutrition and Dietetics, they've all come out with policy statements that by and large support the use of low-calorie sweeteners for those that would like to add it to the regime to control their body weight. But they're not ringing endorsements. They're a bit equivocal. And if you look at the Dietary Guidelines Committee, so they obviously had access to all the meta-analyses, well, not the Canadian one, but the first two meta-analyses as they were published in 14 and 15. So they were aware of that literature, and they concluded that there's moderate and generally consistent evidence based on the RCTs that there is some potential benefit by use of these low-calorie sweeteners, and that it's equivocal based on the cohort studies. But their final conclusion was that the sweetener should not be recommended for use as a primary replacement substitute for added sugars in foods. I think, that's a judgment call. We can interpret the literature different ways on this, but this is kind of where we stand. So I want to spend the rest of my time actually presenting what may be a new concept, but I think a very important one. If we're going to move this field forward, if the reason we can't agree on whether or not these products are efficacious is that the trials aren't adequately powered, they're not run for long enough periods of time, if we're going to be looking at methodological issues, I think there's one that's really been overlooked that may turn out to be critical, and that is we view all of these sweeteners as one common class of chemicals, right? They are all things that are sweet and can be added to food without contributing energy. And the assumption here is that their mode of action is energy dilution, right? You put these in instead of a nutritive sweetener, you have a lower energy product that's matched on sensory properties, close. And so it should be beneficial for those who are trying to manage their body weight. But I think we have to challenge that concept. In fact, and I'll give the punchline right now, these are widely discrepant chemicals. Why would we assume that they all have the same physiological and biological, behavioral responses? Lead acetate certainly doesn't have the same physiological response as aspartame, right? We know that already, but the regulatory bodies, the scientific community, certainly the consumers, we all just, they're low-calorie sweeteners, right? And they're all the same. Let's explore whether that's a valid concept. I'm not going to go into great detail on the basics here, but for those of you not familiar, the sweet receptor is this heterodimer, only discovered, by the way, in 2001. Really a very new concept. And history may show, so the discovery is attributed to Zucker, but history may show that Monell, in fact, the group there may in fact have predated the discovery here. But nevertheless, so we have this heterodimer, and there's some argument that there may be an accessory sweet receptor, but by and large, this one receptor seems to be mediating the sweetness of nearly all the sweeteners that we find in the food supply. This very vast array of chemical structures all being mediated by the same receptor. It seems to violate the sort of specificity of receptor ligand binding sort of things. So how does this happen? Well, they bind to different places on the receptor. So there's sort of three domains. There's the amino acid domain, the venous flytrap, and then there's the cysteine-rich domain in between here, and then the transmembrane domain. And so each of these sweeteners binds to a different place. Now, why is that important? Well, it seems to lead to different intracellular signaling processes. I'm not going to go into the whole cascade of intracellular signaling. Suffice it to say that a rise in calcium is part of that process, as well as a change in cyclic AMP. So here are data on different low-calorie sweeteners. So here's ACE-K, and what you can see is that it leads to a sharp and sustained rise in both cyclic AMP and calcium, with the rise in calcium being a bit greater. But if you look at sucralose, you get, again, a sharp rise and a sustained response. But in this case, the cyclic AMP is a greater response than is the calcium response. If we come down here to saccharin, there's no effect on calcium, but there is the rise in cyclic AMP. And if you go to blanking on glycyrrhizin, what you see is no effect on cyclic AMP, but a rise in calcium. So different mechanisms for all four of these low-calorie sweeteners. What's the implication of that? Well, we know they lead to different sensations, right? And you'll often see figures about how intense, what's the potency of each of these compounds, and you'll see numbers like that. And I just want to point out that this is a ridiculous set of numbers. First off, in your personal experience, have you ever put two things in your mouth that differ in intensity by a factor of 20,000? It's just not in the realm of human experience. But second, the association between concentration and sensation isn't linear, it's curvilinear. So the magnitude of these effects is going to be dependent on where you are on that curve. Suffice it to say, though, that there are clearly differences in the potency of each of these compounds. And they have different sensory profiles. So, for example, ACE-K has a very early onset, but a very short duration. In contrast, neotame, which is popular for chewing gum because you'd like to keep it sweet for a good long time, right, has a long lag phase, but then a gradual rise in sweetness that's maintained for a considerable amount of time. And the others are kind of intermediate here. So these chemicals, these low-calorie sweeteners, all differ in their sensory properties. I might also point out that rodents, are not the best model for studying sweetness for humans. So humans can taste all of these compounds, a number of proteins and cyclamate, aspartame, a dipeptide. We can taste all of these as sweet, but rodents either can't or have a much blunted response to each of these compounds. Rats seem to think that fructose is less sweet than saccharin, than sucrose, and humans are just the reverse. So really quite different sensory functions in rodents versus humans. So it's a question about what we, and of course they metabolize carbohydrates quite differently. Rodents are very efficient at converting excess carbohydrate to fat. Humans, not so much. Okay, another thing that has arisen just in the last decade or so is the recognition that what we have termed taste receptors, because they've been identified first in the oral cavity and subserve taste sensations when the appropriate chemicals are put in the oral cavity, turns out they're all over the place. Sweet ones, bitter ones, but this now raises the question. What we thought was a compound, these low-calorie sweeteners, yes, they were active in the oral cavity, but once we swallowed them, they were pretty much benign. Now the question is, well, is that the case? If it's the same receptor and it's all over the body, what happens when these compounds interact with that receptor, that exact same receptor, elsewhere? What are the effects there? Well, it depends on the tissue in which you find the receptor. So if it's on a taste receptor cell, and an appropriate ligand, low-calorie sweetener, binds to this sweet taste receptor, you generate a neural signal that is conveyed through the gustatory system and is decoded as sweetness. That's the response to it. If, on the other hand, that same receptor is located on an enteroendocrine cell in the intestine, and a sweetener is in that location and can bind to that receptor, instead of a sensation of sweetness, which, thank goodness, we aren't detecting the taste of ingesta as it passes through our GI tract, rather than generating a taste response, it activates the release of gut peptides, so GLP-1, for example. And there are interesting things here. So all the sweeteners are in the mouth. We taste all of them as sweet, but only selected ones manage to get to the intestine. So aspartame, for example, is fully digested in the upper duodenum. It's just not going to get there. It's not going to be effective at activating this pathway